You think we should start?
I think you can go ahead.
Okay. Good morning, everyone. It's a little bit of gloomy morning today here, and we have a special person to present to us today. It's Dr. Fathi Karouia, who right now is pretty much located in the future. Fathi right now is in Korea, and it's 12:30 a.m. there. So we are very, very appreciative that you are here today with us. And at this rainy morning in California, you're going to take us out to the space. So let me introduce him properly. So Dr. Fathi Karouia is a senior research scientist within Blue Marble Space Institute of Science at NASA Ames Research Center and a leading expert in space biology, astronaut health, and performance and space technologies to enable exploration. I have to repeat this again. With over 2 decades of experience, Dr. Karouia has contributed significantly to understanding the molecular and cellular mechanisms influenced by space flight, conducting pioneering research into the biological impacts of microgravity and cosmic radiation. His extensive portfolio includes over 35 successful experiments on board the International Space Station, advancing critical knowledge for long-duration space exploration. Dr. Karouia is also a research faculty member at University of Colorado Boulder, where his work focuses on in-space manufacturing technologies to address pressing health challenges on Earth. Amazing. By leveraging the unique properties of the space environment, he's advancing innovative solutions in biotechnology, such as hematopoietic stem cell expansion for therapeutic applications. Dr. Karouia holds a BS in physics and chemistry, an MS, Master's in Physics from Henry Poincaré University, another MS in Applied Space Technology from Louis Pasteur University, and another Master's in Space Studies from the International Space University, and a PhD in Biology from the University of Houston. Driven by genuine passion for humanity's future as an interdisciplinary species, his research bridges exploration science and tangible benefits for life on Earth. And today he will talk to us about reproduction. Thank you very much and welcome, Fathi.
Thank you. I think I just disappeared for some reason. I don't see myself, so I guess there's an issue.
Daikin should be able to fix it. Oh no, I don't see you too. Just wait.
Maybe it's an issue with, let me remove the background, I guess. Maybe that might work. All right, yeah, I guess back to Earth. Yeah, I'm not sure why. Anyhow, thank you again for the kind introduction, and let me put my slides on. You're able to see my slides, right?
Yes.
Okay. So thanks, Christine, for the kind of introduction. I greatly appreciate the opportunity to be able to speak with you. I know it's early morning in California, so please, I just want to convey that I greatly appreciate the opportunity to speak with you on an early day on Monday. And what I wanted to do in some sense to kind of take you on a brief journey related to reproduction and reproductive health in general. And I know maybe some of you have, in some sense, participated to some of the seminar, webinar that I gave recently. And so I promise certainly there will be some overlap, but I promise also there will be some new material so you don't get bored. And so let's get started. Well, first of all, I wanted to thank obviously Lucine and Lils for obviously the invitation. Again, it is a great opportunity for me to be able to speak to the Stanford family, and in particular OBJ1 department. And so again, thank you. So I wanted to kind of convey a few things. And of course, before I start, as it was alluded into, I mean, I've been involved with NASA for quite some time, 25 years and counting. However, I'm not here as an official representative of NASA. And so all the different topics and in some sense, all the different things I'll be talking about and kind of related to some of the research that I've been conducting for the past decade or so, and again, it will be kind of emphasizing on human health in general and reproduction in particular. And then at the bottom here, there are some of my affiliation here. So let's get started. And so I think some of you, probably or most of you, are not very much familiar with space research. And so I thought it might be important to kind of start to set the stage and kind of tell you a little bit about space environment, and more importantly, about astronauts' health and performance, and then kind of dive into reproductive biology in space. And then at the end, I will be briefly mentioning some operational, clinical, and ethical consideration. So I wanted to start with the following in some sense. I mean, probably many of the individuals on the call maybe were not born in the 60s, but in early 60s, if you don't remember, but there was strong interest from President Kennedy to in some sense compete with the Russians. And then in 61, I mean, there was a key speech which was actually held in Houston and at the Rice University Stadium. And that's more or less where he kind of laid out the foundation for the lunar exploration. And then, so again, this was on 61. And then during Apollo 11, that was the first time that actually we had an individual on the lunar surface. And of course, that was in 69. So within less than a decade, in some sense, I mean, we were able to kind of transition from a wheel to actually be on the lunar surface. And I wanted to convey the fact that for most of you, this is something which is actually relevant. Because actually in 69, again, which was the time where we had the first footprint on the lunar surface, that was more or less the first two papers that came out kind of related to IVF. And then less than a decade later, and here it's a picture of the Cambridge physiologist Robert Edwards holding the first test tubes baby, which is, her name is Louise Jo Brown, which was born in 1978. And here it's a picture of her actually from last year. And I thought that was a kind of interesting parallel between the two, where, early on in the 60s, you had that vision from Kennedy, and then less than a decade later, we had the first footprint on the lunar surface. And similarly, the first paper related to IVF came out in 69, and then less than 10 years later, we had the first baby. And so I thought that was very, very remarkable, and I wanted to emphasize that. So let's get on that journey. And again, this is not the sperm going through the moon, the sun, I'm sorry, but actually it's Falcon 9, which is a spacecraft that NASA uses on a regular basis. And so I wanted to kind of convey the basics in relation to the space environment, because again, many of you are not very much familiar with that. And so when you deal with the space environment, there are a few things that you need to kind of be aware of. Of course, we're in space, where's the outer space, so it is a vacuum. One thing also to be aware of, you have a very large temperature gradient, when you're facing the sun or not. And so temperature can go from 100 and minus 157 to plus 121. So very, very huge gradient from a temperature perspective. And then, of course, there's a gravity that can change. So, of course, when you low Earth orbit, which is kind of more or less where the International Space Station is located, roughly 400 kilometers above Earth, we can experience almost microgravity, so 10 to the minus 5, roughly. So, and that's mainly due to the fact that because it is an habitat and you have devices and instruments on board, therefore there's a little bit of vibration. And then, of course, if at some point we had, and hopefully very soon we'll be heading back to the moon, then that would be more or less 1/6 of Earth gravity. And then hopefully within the next decade or so, we'll be having individuals on another surface, which is roughly 40% of Earth gravity. So, of course, from a gravity perspective, there's a lot of changes that need to be considered when we envision a human spaceflight. And then radiation. And so from a radiation perspective, there are three different elements. But before diving into the specifics related to radiation, how that impacts human physiology and reproductive health in particular, let's kind of go in some sense with some of the basics. And so when you use in terms related to physical properties or biological processes in general, I mean, if you look at G as parameters or variable, there are many, many different equations where the gravity is part of, which means that if that element or parameter change, then all those different properties will be also impacted. And certainly it is something that we do see even here in low Earth orbit, and actually it kind of corresponds to a shift of in some sense how life sciences and physical properties are behaving. So here, for example, there's some images that kind of emphasize the fact that when you here on Earth, where things settle here in this case down to that vessels, when you're in space, there's no sedimentation, and therefore there's properties which is of course very interesting to kind of leverage. Similarly, there's no convection, and so here you see two different phenomena that you can observe. So this is a flame on Earth, this is a flame in space, very, very, very different properties. So when those characteristics decrease because of a lack of gravity, there are other properties that takes over, in particular surface tensions. In this particular picture here, you see the Italian astronaut Samantha Cristoforetti, which love espresso, and actually took a few iterations to design a cup which actually leveraged surface tension to be able to keep the liquid inside the cup. And so that was the only way actually for that espresso to remain in the cup. And so again, there are a few things that that increase when you in space, and those are things that again that you can leverage. But from a biological perspective, those are also a lot of things that would be impacting science and life science in particular. For example, I think I was mixing with my slide, but I apologize for that. So, for example, here you have an interesting paper that can leverage how space and microgravity in particular is impacting life science. In this case, we're looking at some cells, and osteoblastic cells in particular. And so here you have a few different pictures looking at cells, and again, osteoblastic cells under 1G. So 1G is like Earth gravity, or micro G, which is like on board the station, or maybe using some devices to simulate microgravity condition. Well, first of all, if you look at the nucleus, and again here we use DAPI as a fluorescent dye. Well, clearly you see that the overall structure of the nucleus is very different when you compare 1G to microgravity condition. Similarly, when you use that specific biomarker for proliferation, in this case Ki-67, very different profile between the two conditions. But more importantly, when you try to assess, in some sense, the overall structures of a cell, in this case, looking at two things: one, the cytoplasm, as well as the lysosomes. So the cytoplasm here is in blue, where the lysosome is in red. I mean, clearly you can see also strong differences between those two conditions. And so clearly, when you're in space, and again orbiting around Earth, well, those different type of environment, in particular microgravity, will have a tremendous impact on the cell. So at the nucleus level, at the proliferation level, and also at the organization or the structures level. So all those things will impact life. So I wanted to also emphasize that there's a lot of things that can be talked about reproductive health. And here is, the Y and X chromosome. And so I just wanted to emphasize that I will be briefly touching on the male portion, but the emphasis of the talk would be more on the female side. And so again, I just want to emphasize that aspect. And so again, there's issues related to male reproduction, and I will be just briefly touching upon it. But I think based on the audience, I wanted to kind of focus more on the female part. So from a NASA perspective, again, the idea is to kind of leverage what we know from activities in low Earth orbit and the International Space Station. Then at some point, and hopefully within the next few years, heading back to the moon, and then maybe, as I mentioned earlier, going to Mars. And so the Human Research Program, which is an element within NASA, is interested in some sense in understanding the risk and then mitigating them. And so from a risk perspective, which are those small icons right here, there are 5 of them. So one of the main ones, which I'll be talking in the next slide, is related to radiation. Another important one is gravity fields, because again, depending of your location, the gravity level will be different. Then there is isolation. So as you can imagine, you're in a habitat and so far away from family and etc. So that has a tremendous impact on the human physiology. And then you live in a closed habitat, right? And so that habitat in some sense can be considered as a hostile environment. And therefore, there's a lot of consideration in that regard. And then the last one, which is just right here, specifically related to the fact that, when you're starting to kind of move away from Earth, then there's another issue, which is what we call distance to Earth. So I mentioned that radiation, in some sense, is probably one of the main elements and one of the main limitations from exploration, and in particular when we talk about long-duration spaceflight. And so, again, there are 3 main things that you need to understand. One is that Earth, in some sense, is North Pole, South Pole. Because of that, there's a magnetic field. And because of that, then some particles are trapped. So we call those the radiation belt, in particular, Van Allen radiation, which was the first one to discover those belts. Second thing is, we're living close to a star, which is the sun. And so the sun is very, very active. And so there's a lot of eruptions that occur time to time. Luckily, most of them are directed away from Earth, but at times this is directed towards Earth. And so those are also consideration to have. And then the last element is what we call galactic cosmic rays. Those are actually remnant of supernovae explosion, which are coming from outside of our solar system. And certainly those are also things to consider. And those are probably one of the main elements in relation to a long-duration spaceflight from a stochastic perspective. And what is also important to kind of consider when we talk about radiation is the fact that all those radiation elements, so again, galactic cosmic rays coming from the sun or trapped particle, when those interact with matter, they generate single-release particles. And sometimes those single-release particles are more damageful than the primary. So those are considerations to have in relation to the type of shielding that you want to have, maybe having some propulsions and a system that in some sense reduce the exposure to radiation during transit and etc. So all those are considerations to have. From a radiation perspective, so I mean, most of you are physicians, so you go to the clinic to get an X-ray of your chest, so that corresponds to X, and in this particular case, 0.1 millisievert, which is a measure of radiation. Well, if you spend 1 year on the ISS, you will receive roughly either 1,400 X-rays, so similar dose than 1,400, up to 2,500. So it's quite significant. But if you envision a mission to Mars, which will be roughly 6 months to go to Mars, you stay there probably a little bit over, maybe 2 years, maybe a little bit less, 6 months to come back, you will receive a dose which is similar to 12,000 dose of X-ray that you get in a clinical setting. So again, it is not something which is insignificant. From a dose exposure, I mean, of course, astronauts are working for specific agencies, and so therefore we need to monitor their exposure level, and of course there are some limits. And actually, this is a graph coming from one of the documents that emulated from the national academies and can emphasize the fact that for a very long time, actually, NASA was very much on the conservative side in relation to exposure, and on the conservative side to kind of limit the exposure to the crew. But more importantly, because it's well known that body parts of female are more sensitive, radiosensitive than male parts. Actually, those limits for women and female astronauts actually were different compared to male. And in some sense, that could be considered unfair. And so actually, 2 years ago, NASA decided to change its policy. And actually now the dose actually is 600 millisievert. But there's no differences between gender and or age, which actually is very, very different from what it was in the past. But actually, if you compare those 600 millisieverts to other agencies, as you can see, NASA is still on the conservative side. So from a perspective from a NASA approach, and when we deal with exploration, I mean, human factors is an integrative part of the system. And so again, I'll be briefly talking about the impact of space on the human physiology. But as you can imagine, when there is human in the loop, where you need to have systems which are adequate for enabling human to survive. And so from an environmental perspective, from a spacecraft perspective, from extravehicular activities, and etc. All those are things that you need to consider. And again, humans are a fully integrated factor when we envision a long duration spaceflight. When you work in the field of space, well, there are things that can be done on the ground. And ultimately, when you want to do studies, you want to be in space. And so again, it could be space station, or it could be free flyers and etc. But again, it is very complicated. It takes a lot of time to be able to do so. So there are things that you can leverage. So certainly you have some isolation habitat that you can leverage to kind of mimic some aspect of spaceflight, drop towers to kind of do like short-term exposure to a microgravity condition, bed rest studies, which are something that the US as well as European counterpart use on a regular basis to kind of mimic the unloading in particular. Similarly, we use a lot of animal studies. So here it's mice where we actually suspend either through the tail or either through the pelvic. So we call that handling unloading. And then, of course, there are facilities that you can leverage to do radiation exposure. And then there are devices where you can do culture. And so here it's a rotating wall vessel in some sense, which are also used to be able to kind of simulate microgravity conditions. So again, there is like a large playground of devices or environment that you can leverage to be able to better understand the impact of space, or maybe the impact of microgravity, radiation, etc. And so certainly those are things that you can do here on Earth, but again, the mecca will be to be in space. So I promised to kind of briefly talk about human physiology. And again, here you see all the 5 different hazards that I briefly mentioned earlier. And actually, the overall physiology is impacted by the space environment. And again, it's not just microgravity, it's microgravity, it is radiation, of course, and etc. So for example, when you're in space, well, you're not using a muscle, therefore there's muscle atrophy. Usually you lose kind of 5 to 10% of muscle size, up to 20% of muscle strength. Similarly, because, well, you're not putting any mechanical loading on your bone, well, there's also bone atrophy, and so bone loss density roughly 1 to 1.5% per month. And so if you stay 6 months, you might have an impact up to 10% on your skeletal system, which is pretty significant. Other aspects related to cardiovascular deconditioning, other aspects which is something kind of recent, maybe for the past 10, 12 years and 15 years maybe, which is neuroocular syndrome. And so some crew came back with some visual impairment after being in space for quite some time. Still don't understand the reason, probably a combination of redshift, which could be one probably element which is impacting the overall neuroocular system. And then, you immunocompromise. And so when you're in space, all the different key elements related to the immune system, so natural killer cells, T cells, neutrocyte, leukocyte, all of those are impacted, either from an activity perspective or either from a production perspective, and etc. And then actually there have been also many instances of viral reactivation while in space. Of course, at the molecular level, you see all those changes. And so, of course, there's oxidative stress because of radiation, but also because of the stress of mission, stress, the fact that you're isolated, etc. DNA damage, mitochondrial dysregulation, which seems to be a key element related to human in space, and then of course a lot of changes and etc. So again, here's some brief introduction related to some of the key references associated with the impact. So from a NASA perspective, again, NASA is not interested in resolving everything, just interested in going from A to B. And so then either if you do short-term or long-term exploration, then there are some risks. So when it's green, it's okay. Yellow, it's so-so. When it's red, it's a problem. And everything here is kind of related to those elements. And as you can see, there's nothing related to reproduction and reproductive health in particular. Here is an interesting chart that kind of highlight how all the different hazards are kind of connected to one to the other. And so I wanted to kind of highlight that. So again, from a space perspective, this is what we know. So we've been in space for quite some time, since over like 60 years now. And roughly for over a year, again, on the ISS, we've been to the moon shortly, but planning to go back for a longer period of time, which is unknown, and then at some point going to Mars, which is also unknown. So from a reproduction perspective, there have been many studies since the 60s, kind of looking at reproduction in space, using different animal models. And again, for example, here, fish, which was the first vertebrate to be actually reproduce in space, hatch babies in space, and then came back to Earth. And again, there have been a lot of studies related to that. I will be kind of diving into more animal models and mice in particular, and then also some cells. And again, mainly emphasizing currently previous and ongoing studies on the ISS. So I mentioned I will be briefly talking about male, and so this is a study actually I was part of, which is micro 11. And yes, this is Homer riding a sperm bull, and so because we flew sperm as well as bovine sperm. Actually, initially we had a NASA logo there, and then NASA was not happy because they were telling us, well, we're not endorsing Simpsons, so please remove it. So then we just put like a US flag instead. But that was the first time where we were flying as Homer in space. Actually, we had also sperm from sea urchin in an earlier mission, which was actually on the shuttle. The reason I wanted to kind of briefly talk about this particular project is the fact that kind of leverage how complicated it is to do science in space. In the case that I was just trying to convey here, where we flew bovine and human sperm in space, the idea there was to do a few things. One is to kind of assess motility. What is the impact of microgravity? To also assess achromosomal reaction, and again, what is the impact by microgravity, and then trying to see also DNA damage, if any. And so for that, we had two actually astronauts that kind of worked, because it was a very complex mission. And so those two astronauts were Serena Hammond-Chancellor as well as Drew Foster. And the beauty of being in space is that actually, well, you can leverage that the fact that you're in space, and so there's no up and down. And so here, for example, it's Serena. So this is the glove box. So next to it, we had a microscope because, again, we wanted to do some motility assessment. Then, we had a camera, so that enabled us to be able to see what's happening. We had a software which enabled us to do measurement in real time. Then this is our samples where we had the sperm cassette to be able to visualize motility. So we had a timer here. Then the freezer is up there. So you don't have to kind of move around. You just open the door, get the things from the freezer, and then be able to kind of do things, take things into the glove box. And then here is an incubator, which we leverage also to thaw the samples. And so as you see, here it's pretty kind of unique environment, which is not something that you can do when you're in a setting here on Earth. And then, of course, here you're not seeing, but we had some cameras and etc., which enabled us on Earth to be able to monitor the crew's activity. So, and again, it was very complex. And so, for example, we had some media, and then we had the sperm inoculum, which were actually stored at 150 degrees. There's no liquid nitrogen in space. And then we have to thaw those, and then to kind of room temperature, so we can use the bag to do that. So all that is taking place outside the glove box, right? Inside of the glove box, so we take the media, we add it into a culture bag, then we take the sperm sample, we mix it into the culture bag, And then this kind of T0, and then we take an aliquot and put it into the cassette, and then we can do a measurement using the microscope. And then we repeat that at different time points. And then also at the end, we can fix those different samples. And so, again, very, very complicated. And I wanted to kind of show you actually some real-time imaging from, I think it was maybe human or bovine, I don't recall. But nevertheless, those are sperm samples in space, which were actually measured in real time. And so which was pretty amazing. I don't have time to go into the data, but I just want to tell you some of the drawing conclusion. One is that when we're looking at capitation of sperm, I mean, again, we were kind of looking at motility. Well, when we were measuring that and comparing the motility parameters to Earth base, and so again, 1G versus microgravity condition, there is some changes. And actually, those, in some sense, the progressive motility as well as curvilinear velocity, those were impacted by microgravity as compared to Earth, which means that from a motility perspective, microgravity impacts motility. Similarly, and again, I don't have time to kind of go into details related to achromosomal reaction, but when we were doing assessment of achromosomal reaction in space, we saw some differences versus the ground, and differences in a negative way. And so that was a problem. Similarly, I mean, we had sperm stored on the ISS for quite some time, and actually we measured some DNA damage, and DNA damage from the storage, but also DNA damage when we were measuring in real time, which of course, probably that came from the radiation exposure. Here, I wanted to kind of also emphasize the fact that we were on the Daily Show a few years back, and so if you want to have a good laugh, it's only lasts for 2 minutes. I highly recommend to watch that video where Trevor and Noah is kind of making fun of us. So let's shift gears and let's briefly talk about women's health in flight, and actually either ground-based or space flight-based. And here it's a picture of Jessica Meir. And so before I dive into some specifics, of course, women have been in space for a very, very, very long time. Started from the Russian side with Valentina Chergaskova. And again, there have been many different, in some sense, hurdles early on. I mean, as you were aware, it was men-dominated endeavors. Nevertheless, first cohort was in 78. The first woman from the US in space was 83. And then, surely now we see more and more female presence in space. Kate Robbins, for example, was the first one to sequence in space. More importantly, Christine Koch, actually, she is currently the one that has stayed longest in space. So it was 328 days. And actually also during her mission, Jessica Meir joined her. And actually that was the first time ever. There was the first all-female spacewalk with Jessica Meir. Of course, from numbers of days in space, I mean, nobody can beat Peggy Whitson. So she retired a few years back from NASA, but actually now she's part of Axiom. And so now she can fly to the station as a private astronaut, as a chaperone. And then again, the idea there is to have the first woman around the moon and then on the lunar surface, hopefully sometime soon. And actually, Christina Koch hopefully will be launching next year, and she will be the first woman orbiting around moon. One thing that I wanted to highlight, and I wanted to make a tribute to Jessica Meir, which is just right here. And actually, we were classmates. And actually, I was going to go and start a PhD in biology. And so that was my birthday there. So that was quite some time ago. And actually, her gift was to kind of give me a dictionary of biology, nevertheless. And what I wanted to do actually is a tribute to her. And so I do a lot of science in space, as it was mentioned. And so actually, I do also a lot of space microbiology. And so we collected some organisms from the International Space Station, some of which are novel. And so actually, we just published a few days ago, I mean, I guess a little bit over a month ago now. And actually, I just named a microbacterium, which was isolated from the crew quarters on the International Space Station, Microbacterium miriae, as a tribute to my classmate, Jessica Meir. So again, I mentioned that from a space exploration perspective, I mean, when you compare male to a female, it is a tragedy. I mean, to be honest, it is a tragedy. So on this slides, you can see the ratio of astronauts by gender. And so overall, tiny portions of Explorer have been female. But what is also significant is the amount of time that women have spent in space compared to men. And as you can see here, only 2%. So again, if you add all the amount of time that humanity has spent in space, only 2% of that correspond to women or female astronauts, which is a tragedy. And NASA, starting to change that. Now, every time there's a selection, you will see it's not roughly, it's actually half of the candidates are female. And so again, we are now trying to kind of, in some sense, change that, but it will take time to kind of understand how the female physiology and the female reproductive health is behaving in space. And this was noticed from the national academies. And so NASA, and in particular, the space biology program, as well as the human research program, every 10 years ask the national academies to kind of please do a study and tell us what we should be doing in the next decade. And so a document came out in 2023, kind of highlighting what NASA should be doing for the next decade. And here I'm just highlighting a few things related to reproduction and reproductive health in particular. And so one is kind of related to the effect of space on cancer, which of course is very, very important. Others is related to sperm development, ovarian functions, and again, how those are impacted by the space environment. Similarly, how are conception and pregnancy initiated in space, and does the environment affect all those different processes? Similarly, how does prevention of pregnancies and menstrual separations, viral conceptions used by women crew interact with risk of in-flight blood clot? And so again, there have been some studies which have shown that the thrombosis opening in space, which is of course of concern. And then again, going back to the basics about fetal organ development, and then also ovaries, testes, how those are infected by the space environment, microgravity, radiation exposure. So all of those things are things that we need to dive in and work on. And again, you guys are the experts. I mean, I'm not an expert. This is your territory. This is your playground. And that is based on decades and decades and decades of research. Well, everything you know actually might be different, and most likely it is different in space. So all the things that you assume which is happening related to reproduction and reproductive health is actually impacted. We know that for sure, because again, things are changing at the nucleus level, things are changing at the genomic level, things are changing in the overall structural level, things are changing everywhere. So all the things that you do see here might be impacted one way or the other by space environment. And again, microgravity, radiation, and etc. For example, here, if you dive related to kind of space radiation, and again, when you're in space, here, for example, the type of radiation which is coming from galactic cosmic rays. And as you can see, when you compare, for example, gamma radiation to heavy ions like iron or oxygen, I mean, the impact to the cells is pretty significant. So that leads to double-stranded breaks and etc., which are very, very different from a biological perspective to those type of radiation, X-ray, gamma radiation, etc. So there have been a lot of studies related to the impact of radiation. And again, from a damage perspective, there's a direct effect related to reactive oxygen species or the direct effect related to DNA damage. A lot of studies have been done, related to radiation therapy. It is well known that ovaries are very, very radiation sensitive, and certainly there have been some impacts and related to, for example, primary ovary insufficiencies, which is something which is key. And for example, here in rodents, when the ovaries were exposed to oxygen-based ion radiation, there was a primary ovary insufficiencies, which led, of course, to kind of DNA damage. Similarly, I mean, other studies looking at different type of radiation, I've seen some similar outcomes. And again, I mean, these are things that we have known for quite a long time. And again, similarly, I mean, when you're exposing different tissues, in this particular case, ovarian tissues, to heavy ions, and in particular here, iron radiation, can lead to ovarian tumors. And so just wanted to highlight, actually, a few years ago, we published a paper called Extrasurgical Gynecologies. And so here specifically looking at the impact of developing cancer in female astronauts. And so that was published 2 years ago. So as you can imagine, when you're in space, well, you're in space, the ISS orbit Earth every 90 minutes, which means there's 16 sunrise and sunsets. And so that has a tremendous impact on the circadian clock, right? And we know very well that circadian rhythm actually pathways are regulated in organs during spaceflight. Similarly, we know that gravitational changes, either hypergravity or microgravity, can alter circadian clocks. But more importantly, clock genes exist in reproductive organs, right? And all those, for example, are important genes which are important and related to the circadian clock, and those are found in the ovaries. And so, as you can imagine, when you're when you're in space orbiting around Earth, I mean, all those different processes might be impacted just because of that. Then, you add radiation, you add microgravity condition that has a tremendous impact. So this is something which actually derived from some early studies, and here are the different references. And so again, here using symmetrical microgravity condition, when there was some male rats which were exposed to the environment, and here is kind of specifically located at the gonadotropin releasing hormones, as well as the hypothalamic pituitary gonadal axis. And again, if you look at all those different genes, genes which are related either within the hypothalamus, or those with LH, so levitating hormone, or the follicle stimulating hormones, where those are impacted and negatively impacted. Similarly, similar studies looking at radiation using iron as well as oxygen on female mice. Again, there was changes related to FSH as well as LH. And in flight, again, there have been also some study, not much, but there have been some studies which actually saw some changes, but some of which actually didn't see no effects on the pituitary as well as the ovarian mass, which was a good thing to see. From a menstrual cycle perspective, again, there have been some interesting studies specifically looking at some of those changes. For example, in mice, there was some cycling which actually stopped, the estrogens in particular, and also some changes that were observed. When we use ground-based studies, again, the Himlin suspension, so for specific rats, again, we see also some changes in the cycle length. And there was a study which were actually done in space, and that was using mice as a model organism. And so here actually you can see some results looking at the different stages of the menstrual cycle. And when you compare flight to a baseline or to the vivarium, I mean, there were some changes. And so all the mice were in different stages of the menstrual cycles as compared to ground-based counterpart. Similarly, I mean, you don't see differences in E2, which is the estradiol. However, there was some significant changes related to progesterone, P4, when you compare flight versus ground-based. And so, again, some changes, but there was no differences in the overall ovarian tissue E2, as I just alluded into. From an ovarian folliculogenesis and steroidogenesis, a few studies here that I want to highlight. Again, it's either using symmetrical microgravity condition or spaceflight. In these particular cases, they were looking at specifically some rate of oocyte maturation, and actually they saw there was a decrease rate, and actually pretty significant. We were comparing microgravity conditions, so roughly less than 10% versus over 73% in one gene. Similarly, there were some changes in the overall structure, some disruption in mitotic spindles, which was seen. Also, some changes in the tubuline. And again, that kind of go back to the story what I was mentioning earlier, where there's an impact of microgravity condition overall at the cellular level, but also within the cytoskeleton. Here it's another study looking at some follicle survivors. And so again, there was a decrease of follicle survivors. And there was also a decrease in GDF9 expression, which, as you know, it's a key expression marker. And then again, some abnormal oocyte structures were observed. And so certainly, which was also something to kind of look at. Again, that was symmetric microgravity conditions, so still need to be seen from a flight perspective. However, there was also a flight which was also using mice models here, and in particular looking at specific different biomarkers. And here you can see, so those specific biomarkers, when we look flight versus the others, there was no significant changes. So no significant changes in the ovarian tissue stereogenetic gene expression, and similarly, no significant differences in all those other specific biomarkers there, which was also nice to see. From an oocyte perspective, again, mainly based on ground-based, so again, symmetric microgravity condition. There were some studies that, in particular here in 2021, that kind of emphasized the decrease of proliferation of granulosa cells. Also, there were some changes in the ratio from G0, G1 versus S or versus GA2 M phases. So again, depending on the phases where you're at, some changes were observed when you were comparing symmetric microgravity condition to Earth-based condition. And also some downregulation of specific biomarkers here and genes in particular, but no changes, some changes in morphology. Another study here looking at some, again, it was 2023, kind of demonstrated there was a decrease of polar body release in mice oocytes. Also, it was observed there was a higher level of reactive oxygen species, and then also some changes at the organization level. Here it's looking at some other type of cells, and similarly, it was observed there was some structural abnormalities, decreased aggregates between mitochondria, SRL, large MF complex, asymmetries in the expression of the PV space, and then also alteration in some of the organelles. And so again, despite the fact that those were ground-based studies, I mean, you can see there's tremendous changes either at the cellular level, in the overall architectural level, or at the gene level. From embryo development, again, those were a ground-based study. I mean, when we're looking at embryo development in under symmetric microgravity condition, there was a decreased numbers of embryos reaching the morula and the blastocyte stages after a few days of culture. Similarly, when we look at another study coming from Japanese side, 88% of the mice osteocytes fertilized. However, there was decrease of blastocytes when we're comparing space versus ground. And again, there was also a lower birth rate from those cells. Here we were looking also at cells kind of developing from 2 stages to 8 cell stages. And so actually under microgravity condition, again, here it's simulated, we saw that there was most lethal at 8 cell stages compared to 2 cells. And similarly, there was, it sounds like some key stress-related protein were increased in space. This study was interesting because it was kind of looking also things at the gene expression level. And then so there were some genes which were significantly expressed. And similarly, that kind of led to some changes in some specific pathways, which are also relevant to reproduction health. What was interesting is that there have been some studies looking also at other aspects in embryo development, but more importantly in space. So earlier it was ground-based, those were in space. So in the early days, there was some early attempt, and that didn't work, most likely due to issues with hardware. However, there have been some tremendous work, and probably that derived from IVF technology that you guys developed on the ground. Nevertheless, now you have some systems that actually are flight qualified, and which are embryo culture incubators, which also are automated and actually enables you to kind of monitor the cells in real time. And so there have been studies in the early days where we were looking at the mouse embryo incubation, and actually those didn't develop to blastocytes. More recently, there are some embryos which are actually at the 2-cell stage, and again, those were able to go to and develop to blastocytes. But again, there was a reduction of rate when you compare it to spaceflight versus ground. And so again, tremendous. It seems there's tremendous impact of microgravity as compared to Earth-based. And similarly, as I mentioned, so decreased numbers of cells, but then also there was some differential changes, and then also it was observed there was an increased rate of DNA damage. This was interesting because actually this was looking specifically at means that kind of, so of course a lot of things have been learned from IVF on Earth, but as you can know, I mean, in space things are, you have a limited access to resources, right? And so again, you don't have liquid nitrogen, so you need to come up with new ways in some sense to be able to preserve samples, and in particular a frozen embryo. And so here's a Japanese team actually kind of developed a new type of vitrification to kind of be able to kind of froze embryos at -80 instead of -196 for liquid nitrogen. And actually, you know, they were able to do so. I mean, certainly there was variation in the rate of embryo being recovered, but certainly it was not bad. And actually, those were thought. I mean, many of which actually developed to blastocyte, which was pretty nice to see. This is an interesting experiment, and again, from the Japanese. So Japanese seems to be on the forefront in relation to reproductive health, coming from animal models. And so this was actually 2 cells embryo which were flown to the ISS. So and so actually on the ISS you have a centrifuge. So actually you can also create artificial 1G, which is a very actually pretty unique control as compared to 1G on Earth, because by having a centrifuge in space, actually your samples are mimicking all the different stages, because when you compare space to ground, well, the ground samples didn't go to space. And so when you go in space, there are things happening. Of course, there is acceleration, then you transit to the International Space Station, etc., where those are real control, because then everything is equals, but you only change the gravity level. And so that's pretty cool and pretty unique type of facility, which is currently available on board the International Space Station. And again, those were 2 cells type embryo, and then again, very complex. But the idea there was to kind of having ground control, having 1G in space, and having microgravity in space, grow them for a few days, and then do a few things on the ground. So kind of look at the blastocytes rate, look at cell number, DNA damage, and then gene expression. And again, so when you compare the blastocyte rates to kind of space versus ground, well, there was some changes and kind of still significant, 60 versus 24. There were some changes in cell numbers, but more importantly, when you look at the differentiations, I mean, there was no significance. It was observed there was some DNA damage, and then again, they did some analysis at the genomic level, and in particular, RNA-seq. They did see some differences in gene expression, but again, only just a small numbers, and so, which was nice to see. From a STEM perspective, this is also a new era, so there's a lot of work related to that. And so here I'm just kind of highlighting a few studies which were done either ground-based or in flight. And again, so when you do so, and here looking at embryonic stem cells, and so those can be cultured, which was nice to see, and they kind of maintain their pluripotency, which is kind of course, very important. Here, looking at mice embryonic stem cells, looking at, again, here a study in space, it seems that there was reduced numbers of cells and also decreased adhesion rate. Similarly, there was some delayed DNA repair processes and increased apoptosis, so some concern here on that particular site. Another spaceflight study looking at mice embryonic stem cells. Here also, they can emphasize the inhibit terminal differentiation of those embryo bodies. And so again, this is something which is important. And certainly it seems like microgravity has an impact on the development of those type of cells. And here I want to also to kind of look at that paper, which was in 2019, looking at just pluripotent stem cells, which were also in spaceflight. So those are more interesting than the ground-based. And actually, there was a greater proliferation activity, and actually, it's kind of interesting to see that, and strong OCT4 expression, which, as you know, it's also very, very important gene in relation to embryonic stem cells renewal and differentiation. In relation to endometrium, and so again here a few things. Those two studies are flight-based. This is a simulated microgravity condition. And again, if you look at the uterus and some specific signaling pathways, those were actually impacted microgravity and actually downregulated. And similarly, there were also subsequent studies kind of looking at the uterus, and actually there was observed there was a thicker myosin layer on the flight group as compared to ground-based. Here is Earth-based looking at some stroma cells. And again, there was some changes from a proliferation level, as well as the migration of those cells. And again, decrease of activities of key elements related to the endometrium. So again, here are some emphasis related to some flight as well as ground-based studies, which of course tells you in some sense that space in general has a tremendous impact in the overall physiology. One thing I wanted to mention in the next few minutes, and I will be done shortly, is the fact that you can leverage open science. And so NASA, in some sense, is pretty good in relation to enable everyone to be able to leverage what NASA has been doing. And here I just want to emphasize two things. One is GeneLab. And again, GeneLab, in some sense, is a repository where you have previously Earth-based or flight-based investigation, and anybody can have access to the data. And the other one is biospecimen. So when NASA sends mice in space, and maybe the PI is interested in the muscles and the bone, where all the other organs are dissected, and then they are preserved. And actually, you can request for free access to those samples, assuming that you want to test an interesting hypothesis, and assuming also you have funding to be able to do the analysis. So I just wanted to emphasize that. And there are different working groups. And so those are, in some sense, people which volunteer their time to work. And so currently there are 9 different analysis working group, and I just want to emphasize there's a female reproduction working group that actually Begum Matlik is the chair, and so we meet on a monthly basis on Wednesday. And the idea there, people come with some ideas, and then they recruit individuals to be able to work on some specific hypothesis by leveraging open science. And so in the next few minutes, I want to highlight that. So for example, here we were just looking at specifically at 2 things, which is insulin and estrogen. And so we leverage open sciences. So there was data related to mice flown in space, and we had access to data from all those different organs, but also we had data from astronauts, private as well as professional astronauts that were flown in space. And then we were looking at, okay, well, maybe we can look at specific genes related to insulin signaling or estrogen signaling and insulin resistance. And do we see changes? And actually we did. So here I just want to emphasize briefly, and again, I will be done in a few minutes, a few things. One of which is that we had access to astronauts. Again, those are professional astronauts that were on the International Space Station for like a few months. And again, it looks complicated, but it's pretty straightforward. So in this graph here, we're looking at estrogen-based signaling. So what is in kind of bluish is downregulated. What is red is upregulated. And here there are 3 different data points. Prior, which is this row, during flight. So during flight, there was 4 different time points. And post-flight, there is 4 different time points. As you can see, there's drastic changes. And so here's a list of all different genes related to estrogen pathways. During flight, those were downregulated. Upon return, those were upregulated. It's pretty significant difference. When you look at those specific genes and try to relate to what particular pathway they are involved with, then you can do what we call gene set enrichment analysis. And so here is all those different pathways which are significantly enriched, some of which are related to hormone synthesis, estrogen signaling pathways, ovarian steroidogenesis, steroid hormone biosynthesis. So as you can see here, clearly there is a significant impact of space on all those different pathways. Similarly, here we see the opposite. And again, those are different type of genes related to the estrogen signaling pathways. In this case, those were upregulated during flight and then downregulated upon return. And again, dramatic changes here. Similarly, when you look at gene set enrichment analysis, some specific pathways related to thyroid hormone signaling, for example, here, insulin resistance signaling, which is seen here, and then cyclic AMP signaling pathway, RAP1 signaling pathway. I mean, all those are very, very important signaling pathway. And so again, significantly changed just by looking. And again, here we didn't have access to those to kind of specific organs, the reproductive organ sample. We were just looking at plasma cell-free RNA. And so those were blood-based samples, but nevertheless, we were still able to see those drastic differences, which tells you that by leveraging open science, there are means for us to be able to better understand how the human reproductive system behave in space. And so conclusion, I mean, as I was just emphasizing, embryo development is possible in space. There are many been studies being done in space. However, I mean, again, it's still the field is still at its infancies. And so still more work is needed. As I was highlighted into it earlier with the decadal survey, there's a lot of work that needs to be done. And actually, we need your help. We need your help to kind of address all those things. Because again, everything you know about the reproductive system, everything you know about it, might be questioned by just being in space. And again, specific pathway, it could be at the cellular level, it could be at the organ level, it could be of hormonal level, all those things change. And so we have to have a better understanding, because when we look at long duration space flight, all those things need to be understood. All right, few slides related to operational and clinical consideration. So when you, let's say you're a female astronaut and you just get selected. So let's say you apply. From the time you apply to the time you're selected, it will take roughly 1.5 years. Then, if you're a lucky one, like the one which was just announced like 2 months ago, it will take 2 years to train. Then you wait to be able to assign to a mission that take between 2 to 5 years, maybe sometime more than that. Then when you assign a mission, then you need to prepare for a mission. It might take 1.5 to 2 years to prepare for the mission. Then you go in space, and sometimes because there's some delay, so it might take, you're lucky within the 6 months you're in space, but sometimes you might wait for a very, very long time. I mean, if you combine all those things, those are 11 years in the rest of your life that you need to, in some sense, not be able to have a child. Because again, if you do, then you disqualify from being in space. And so very, very often, I mean, women and female astronauts will delay having a child up until their first mission, and then they try to have a baby. But that has a significant impact on the overall aspect from a reproductive perspective. And so, again, it's very important to have a counseling. And so the flight surgeons, there's individuals like you, and in some sense, those need to be part of the conversation early on, because they need to assess what is the risk from an individual, and in particular a woman that goes to space. And what you need to do is like, okay, well, I need to do an assessment. And so probably doing an ovarian reserve assessment would be critical. And then you need to have a conversation. I mean, should you, in some sense, preserve your oocytes, which most likely will be something that I would recommend. Similarly, I mean, what are the things that you want to do pre-training, pre-flight, and etc. And then, what are the long-term considerations? So all those things are things that you need to consider. And having a talented individual like you guys part of the process is key. Similarly, I mean, as you know, moving forward, we want maybe at some point, that had been on the news for a very, very long time. So yes, they will have some hotel, and for sure sex will be part of that. Certainly, I mean, okay, it's time to take reproduction in space seriously. Yeah, for sure. And then the question, can you even have babies on Mars, and etc. All those things are things that we'll have at some point to address. And certainly this currently is a big elephant in the room. And so what are the ethical considerations? So actually we published a paper last year kind of specifically related to what are the ethical considerations from individuals which are not professional astronauts. So those are private individuals, and certainly very different from professional astronauts, which are the best of the best, healthy, extremely talented and etc. Here are individuals that might have some pre-existing condition. And so are we technically clear to launch? Those are important considerations to have. Similarly, from an ethical perspective, I mean, when you look at missions, for example, going to the moon and Mars, well, should we regulate conception pre or during flight? Well, those are important questions, and even more so from a private perspective, because private entities, private companies might have different rationales. Those could be commercially driven, as opposed to currently the setting from all those different agencies, NASA included, which is more or less going by the book. But similarly, what protection must be in place for those individuals? Similarly, should long-term commercial flights encourage the pre-flight banking of gametes or embryo? Who bear responsibilities for adverse outcome? If medical abortions or emergency deliveries is needed, are these even possible on a spacecraft? I mean, again, when you're under microgravity condition, everything is changing. Now, let's envision like maybe 20, 30, or 40 years from now, we might have some semi-permanent settlements in other worlds and maybe on Mars, for example. Well, what are the long-term rights of children born off Earth? No. What are the autonomy? What are the autonomy of the settlers under contractual obligations? Those are important questions to have, because maybe those are professional-driven missions, but they might be also commercially driven, and therefore their obligation might be different. What about whether and when it's morally permissible to attempt such a premature birth? I mean, those are important questions. And then, what are the implications of rearing offspring in an environment that might make their body ill-suited to kind of return to Earth? So let's say you have a baby on Mars, where Mars is 40% of Earth gravity, right? And the baby kind of evolve on Mars. I mean, can the baby come back to Earth? I mean, how? We don't know, right? Because Earth gravity is very, very, very different. So all those things are things to consider. So bottom line, I want to say is that, we need you, because again, you guys are the expert. I'm not an expert. But there's still a lot of things that need to be learned, even more so from the female reproductive perspective. And then, I would be happy to take any questions.
Thank you very much, Fathi. This is amazing and probably even more expanded than what I heard before. So this is great. We did lose half of people because we went over time, but I would want to pose a couple questions. I don't even know if people who ask them are here, but there are a couple questions there. So Dr. Gibbs was saying, thank you. Please tell us about pregnancy outcomes, including fetal anomalies after astronauts return from their missions. Do we know if astronauts got pregnant and, or male, female, if they had kids, and any pregnancy outcomes with fetal anomalies? Do you know about it?
Yeah, actually, so I talked about two individuals during my presentation. One, Jessica Meir, which is my classmate, and the other one is Serena Chancellor. And actually, both of them had babies after their first mission. And so I don't know the details about, if it was assisted or not, but certainly, I was happy to see. Yeah.
Okay, good. On the Dr. Ames is asking, on the macrophysiology level, have there been alternations to the menstrual cycle among long-term female astronauts? They are all on suppression. My understanding is, due to inability to deal with the menstrual, stuff in the space, they're all on suppression. But when they come back, are there any changes?
Yeah, I don't know. I cannot answer that question. It's when you deal with astronauts and within NASA, then it's a vault in some sense. And so there's not many information related to the overall impact of current astronauts in flight, and what is the impact of space environment on their reproductive health. And I think that's certainly lacking. And so certainly we need your help in some sense to kind of address those gaps. And so I'm not aware of anything related to that. But again, that doesn't mean necessarily that it's not out there.
And then another question is the embryos from space, are they eventually destroyed? And when you do these experiments with the embryos, etc., are there any government funds involved, or this is all kind of private funding?
Oh yeah, I know. Yeah, most of them are funded by governmental-based agencies, and so most of the studies I was emphasizing here are coming from the Japanese side, which is a key partner within the International Space Station. So those guys seems to be on the forefront in relation to reproduction and reproductive health, in particular, using animal models. But yeah, all those are agency-based funding.
That's pretty cool. And the embryos usually are being destroyed, right? Unless
I'm not sure. I'm not sure. I would imagine not. I mean, I think most likely. I mean, again, if it was NASA, so then again you have access to those samples because now if you're funded by NASA, it is a requirement that yeah, it is a requirement that you maintain those samples, and then also you in some sense you should provide those tissues that are remaining back to NASA so those can be distributed as needed, assuming this is NASA. But again, those were a Japanese based funding, and so I think that kind of purview based on their own requirement. But yeah, maybe one thing would be to kind of inquire those PIs to see, assuming someone wants to kind of go back to those samples.
look at that. Another question from Liesel. How hard is it to recreate microgravity conditions for embryo culture accurately on Earth? Is it easy to do or not?
I mean, yeah, there have been a lot of studies specifically related to that, and so there are different systems that you can leverage. In particular, I mean, there's commercially available systems that which are not expensive that you can have in the laboratory to kind of simulate the space environment. And so yes, those. I mean, I've not done the work, but again, I mean, you guys are the experts, so I'm pretty sure that it would be pretty straightforward for you guys to kind of translate whatever protocol you use in a normal setting and just add either a rotating wall vessels or a random positioning machine to kind of simulate microgravity conditions.
Yeah, we can talk about this later if we need to. And then I have one last question. This concerted, so the efforts to help astronauts preserve their fertility, we're talking about maybe egg freezing, sperm freezing. Are you aware that these are the concerted organized efforts, or is somebody, per protocol consulting those astronauts?
Yeah, I did ask the question, and I think it isn't clear. It seems, I mean, certainly. So, I mean, every time there's a mission, the crew is monitored by a flight surgeon, which would be working hand to hand with the individuals. And as I was alluded into, I mean, certainly reproductive health is a key element for the well-being of the astronauts. And so I know in Houston, I mean, in particular, I think it's Baylor College of Medicine kind of working closely, as well as UTMB kind of working closely with NASA. It is my understanding that there are procedures in place. However, I think it's more individual based as opposed to, hey, this is what NASA wants, and etc. And so I think it still remain at the individual level, and in some sense their own journey to kind of seek the right type of counseling to be able to take care of their reproductive health.
Yeah, so there is still space for us to help astronauts.
I think so, yeah.
Yeah. Thank you very, very much, Fathi. I know it's really late for you, so
No, it's okay, and apologies for taking, I guess, all the time. I know
it's
that
okay. You had so much to say.
people
Everybody
I
was
know that
saying, yeah,
you
no,
guys are physician, and so you need to get to your patients, and so. But yeah, thanks again for the opportunity. I know I cover a lot of material, and certainly I would be happy and to come down to Stanford sometime soon and kind of sit down and trying to see how we could collaborate. I know that Liz also wanted to do so, so I would be more than happy to do so. Maybe early next year. That'll be wonderful.
Perfect. We know we have now solid contact with you, so we will definitely talk about this. We are interested. Thank you very much.
Okay, thank you, everyone. Thank you. Bye-bye.
Thanks, everyone.